Okay, good day, students. This is our recorded lecture for exercise number 34, or blood. You will also see in my list of videos that I decided to record the entire cardiovascular system in order for you to have a resource that you can go back to in your own time and on your own pace. Please know that my reference for the entire lecture are both Patton and Seeley. So for exercise number 34, we'll be talking about blood. So if you are ready, let's start. All right. As mentioned before, blood is actually a connective tissue. And in connective tissues, it is a mixture of both cells and a matrix, or the background of the connective tissue. We've already discussed a few connective tissues before, and if you remember, their matrix are composed of protein, it can be composed of ground substance, etc. And together with the matrix are a collection of cells, no? So blood is somehow similar to that, except that the matrix of blood is actually fluid. Okay? The matrix of blood is actually fluid, and the goal of blood is to be able to circulate within the cardiovascular system. That fluid matrix is called plasma. I want you to remember that word. The fluid matrix of blood is called plasma, while the cells are called blood cells. No? The formed elements or the solid structures found in the blood are called blood cells. And these blood cells can be divided into three main categories. You have your white blood cells, you have your red blood cells, and you have the platelets, which we will discuss individually as we go on with the discussion. For females, they have a total blood volume of 4 to 5 liters, while males will have 5 to 6 liters of blood. You may have seen a Filipino teleserye or maybe a movie where they said that the patient is losing blood. This only means that the patient may have total blood volume, which is less than what you see on your screens right now. It's also important to know that the total blood volume actually makes up about 8% of the total body weight. Okay, so around 8 to 10% of your current weight right now can be attributed to your blood volume. All right. Now, when it comes to the composition of blood, there are two main composition. Of course, we have the cells or the formed elements and the fluid matrix called plasma. Let's go and discuss plasma first. Plasma is 91% water, which makes it or gives it the fluid composition and allows it to be able to circulate within the cardiovascular system. Again, 91% of your plasma is composed of water. 7% is composed of proteins. Now, the proteins found in the blood are not similar to the proteins which we have previously discussed when we talked about the connective tissues. Diba? When we discussed connective tissues, we talked about elastin and collagen. But the proteins found in blood are actually dissolved proteins. They are dissolved and they are mixed in the fluid portion. Okay? And the most abundant protein in the blood is called albumin. Okay, take note of that. Albumin is the most abundant protein in the blood, followed by your globulin, and the last is fibrinogen with 4%. Now, fibrinogen, although it is the least in amount of protein, it also plays a big factor because fibrinogen is actually a clotting factor, meaning once fibrinogen is activated, it will cause the blood to clot or to solidify or to coagulate. Again, fibrinogen is a clotting factor, and once fibrinogen is activated in the blood, it will cause it to solidify, to clot, or to coagulate. Okay? So once we collect the specimen of blood, if fibrinogen is not activated, meaning the blood is not clotted, then the fluid portion is called plasma. But if a chemical is added onto the specimen that will cause fibrinogen to be activated, then the fluid portion is no longer called plasma. It is called serum. Again, when the fluid portion of the blood has activated fibrinogen, or the blood has already clotted, coagulated, and solidified, the fluid portion is no longer called plasma. Instead, it is called serum. Okay? So to make things easier, when the specimen is unclotted blood, the fluid portion is called plasma because fibrinogen is not activated. But when your specimen is clotted or is coagulated, it means that fibrinogen has already been activated, and serum is what we call the fluid portion. 2% of those plasma is composed of your gases, such as your carbon dioxide and oxygen, your other ions and nutrients, such as your sodium and potassium. While 45% of your blood specimen is composed of your formed elements, meaning your blood cells. So you have here your platelets, your white blood cells, and your red blood cells, which we will discuss starting with the red blood cells. So red blood cells are also called erythrocytes, all right? It's very important. Red blood cells are also called erythrocytes, and they are the most abundant formed element out of the three. In fact, they compose 95% of your total formed elements. Okay? 95% of the cells found in the blood are actually red blood cells. In appearance, they are flexible disc-shaped cells. If you know what a frisbee looks like, it's somehow similar to how a frisbee appears to be. And the unique thing about red blood cells is that they contain hemoglobin. Okay? They contain hemoglobin, which has two functions. Kindly remember this. The first function of hemoglobin is it is responsible for the red color of the blood. It is responsible for the red color of the blood. And the second function of hemoglobin is to carry oxygen, because oxygen is attached to hemoglobin, and it allows red blood cells to carry oxygen with it. Okay? So hemoglobin again has two functions. It is responsible for the red color of the blood. And second, it actually is the one which carries oxygen in order for the red blood cells to deliver oxygen to the different body parts. So this is how erythrocytes look like. This is just a fun image for us, no? So they appear spherical, they appear disc-shaped, and there is a slight indentation in the middle. If you can see, no, parang may depression in the middle of those red blood cells. Let's now go to your white blood cells. So white blood cells are also called leukocytes, okay? Leukocytes. And their main function is to protect the body against microorganisms, meaning they are our soldiers, our frontliners, no, if you may add. So they are the soldiers of the body which fight off the different microorganisms, whether they be bacteria, virus, parasite, okay? And aside from killing those microorganisms, they also actually are responsible for removing these dead cells by eating them. Because remember that phagocytosis means cell eating. So after the white blood cells kill the microorganisms, they also proceed to eating them and digesting them by phagocytosis. So there are two classifications of our white blood cells. We have our granulocytes, from the root word granule. Why? Because under observation on a light microscope, you will appreciate large cytoplasmic granules inside the cells. And the second classification are agranulocytes. Of course, it has the prefix a, which means the absence of. So it is cells which where the granules are absent. So absent granules. So actually, in truth, they still have cytoplasmic granules, except that they cannot be appreciated under the light microscope. Therefore, we label them as agranulocytes. And here are the different types of white blood cells. By observation, you would also note that they are larger than your RBC or than your red blood cells. So there are three granulocytes. Those are your neutrophils, eosinophils, and basophils. Okay? So para madaling matandaan or in order for you to remember, that's B-E-N, Ben. Ben has granules, basophils, eosinophils, and neutrophils. And you only have two agranulocytes. Those are your lymphocytes, the smallest WBC, and your monocyte, the largest WBC. Now, let's discuss briefly each one of these white blood cells. So, these white blood cells start off with the most abundant, which is neutrophils. This is the one at the end of the image here. Okay, neutrophils, can you see it? All right, anyway, neutrophils at the end here are the most abundant at 60 to 70%. And they actually are multi-lobed granulocytes. If you look at the nucleus of the cells, it has many lobes, right? There's one lobe here, another lobe here, another lobe here at the bottom, and there are actually two more. Unfortunately, my video feed is covering those other two lobes. So usually they have 3 to 5 lobes of nuclei, and the granules inside the neutrophils are actually colored purple, sometimes pink appearing. But what's most important about neutrophils is that they increase during bacterial infection. Meaning when you get infected with any type of bacteria, neutrophils are the specific type of white blood cells which fight off this bacteria. Okay? Next, we have your lymphocytes. Lymphocytes are the first cell that you see in this image. Again, it is the smallest WBC, and it is an agranulocyte. As seen in the definition, it is responsible for antibody reactions, meaning lymphocytes actually produce antibody. And what sickness nowadays talk about antibody reactions? It's COVID-19. So lymphocytes increase during viral infections. Again, lymphocytes increase during viral infections. And in order to fight off these viruses, they actually produce antibodies. All right? Next, we have your monocytes. Monocytes, again, are the largest of all white blood cells, and they are commonly seen in chronic infections. What's unique about monocytes is that they can also be found in the tissues, meaning they can go out of the circulatory system, they can go out of the blood and actually travel to your tissues. But when they get there, they change their identity from monocyte to macrophages. So basically, monocyte and macrophage are the same cell. The only difference is their location. Monocytes are found in the blood, while macrophages are found in the tissues. And again, they are increased during chronic infections. The fourth one is your eosinophils. Eosinophils, 2 to 4%. And they are really unique because they have the orange to red appearing granulocyte, right? What's also unique about eosinophils are they are usually composed of only 2 lobes of the nucleus, no? So there are only 2 lobes here of that nuclei. And usually they appear purplish to reddish to orangish in some specimen. But what's most important about them is that they increase during allergic reactions. So when you eat your peanuts or fish, seafood, or actually any food that you're allergic to, you expect your eosinophils to increase in number. And last but not the least, you have your basophils. It is the least common of all white blood cells, And something that's unique about it is that it has a dark blue to dark violet and sometimes even to black granules. In fact, the granules are so dark stained that sometimes you're not able to appreciate the nucleus anymore. Okay? They appear so darkly stained that you're not able to appreciate the nuclei anymore. Anyway, they're composed of 0.5 to 1% only. Last major type of formed elements are your platelets. They are also called thrombocytes. These are actually the white spined structures that you see here beside your erythrocytes. And they appear like that because they are fragments of a giant cell called megakaryocyte. So here we have a megakaryocyte, and after achieving full maturation, the cytoplasm of the megakaryocyte will start to fragment. And those fragments are called platelets. All right? Now, what's a function of platelets? Their function is to close and repair wounds by forming a platelet plug. I'm sure every one of us has already experienced sustaining a wound in our body. And I'm sure that you've noticed that after a few minutes or after a few hours of from having or sustaining the wound, you'll start to notice a scab, no, a scab on top of that wound, and that scab is actually a platelet plug, which prevents the wound from continuously bleeding. The other name for that platelet plug is thrombus. That's why the other name for platelets is thrombocytes, because the goal of platelets is to produce a thrombus, a platelet plug, to close and repair the wound. Now, putting everything together, this is how a blood smear would look like. So this will be the image that will welcome you every day of your life if you become a medical technologist. No? So let's start to identify some cells here. This cell right here appears to be spherical. We can see a light center, a light appearing center, which could be an indentation or a depression. By the size of it, other cells, such as these ones here, appear larger than it. And there are also some cells in this image which appear smaller, right? So what could this cell be? This cell, which is seen in majority of this slide, is your RBC or erythrocytes or red blood cells. So that is how a red blood cell looks like. Next cell here, this is larger than your RBC. So definitely it will be a white blood cell. Now the problem is there are 5 different types of white blood cells. So what could this be? First, let's classify. Is it a granulocyte or an agranulocyte? Just by observing the appearance of the cytoplasm, you would be able to appreciate this small speckles here in the cytoplasm. And yes, this is definitely a granulocyte. Now, we are left with 3 choices: neutrophils, eosinophils, and basophils. Based on the definitions that we know, a neutrophil is multi-lobed. It is, the granules are purplish to pinkish, and they increase during bacterial infection. Eosinophils, on the other hand, are bilobed. They appear reddish to purplish to orangey. But definitely in this cell, it's not bilobed. It has actually 4 lobes here, right? And basophils have dark staining granules, which we don't see in this image. So, by principle of elimination, we are left with one choice only. This is a neutrophil. Okay? There are 1, 2, 3, 4 lobes of the nuclei. It is granulated. Therefore, this is a neutrophil. How about this cell over here? Definitely, again, this is a WBC or a white blood cell. We can see that the nuclei is bilobed. There are 2 lobes of the nucleus, and the granules are purplish to pinkish to bluish. Um, so definitely not a neutrophil because there are only two lobes of the nuclei. Will it be a basophil? I don't think so because the granules are too light staining if for it to be a basophil. So we are left with one choice and the correct answer is neutrophils. The correct answer is eosinophils. Okay, that's your eosinophil. Bilobe, purplish to reddish to orangey colored granules. Next, we have this cell. And this cell is, again, a white blood cell, no? It has a very scanty or very minimal amount of cytoplasm. So, could this be a monocyte or a lymphocyte? By size alone, we can say that it is a lymphocyte, okay? Monocytes usually have a lot of cytoplasm. But in this cell, it has a minimal amount of cytoplasm. So it can only be a lymphocyte. And lymphocytes increase during viral infections or for antibody production. Next cell is this small speckle here. Again, the smallest cell you will find in a blood smear is none other than your platelets. Again, platelets are fragments of cells coming from your megakaryocyte, which is a giant cell. Now, just to challenge you a bit, would you be able to identify this cell? In other sections, they said that this could be a basophil. Some sections would say a dead RBC, no? But actually, this is still an erythrocyte or a red blood cell. The only thing why it looks like this is that they are stacked one on top of another. So sometimes in preparing your blood smear, this can really happen. Cells lying one on top of another. Actually, this is not the only example we have here. You also have this group of cells here, and this, and you also have it here, no? They can sometimes be stacked one on top of another, but still they are RBCs. Now, blood typing is one of the most common procedures that we do for the blood, right? And it's important for you to know that blood typing is based on the interaction of antigen and antibody on the surface of RBC. This means that each red blood cell, as you saw in the prior image, each one of those cells have antigen attached on the surface of RBC. Again, antigens are present and are attached on the surface of each individual RBC. And this antigen give the RBC its identity. It give it its blood type. Okay? However, as mentioned, however, there are also proteins found in the blood plasma and they are called antibody. Okay? So if these circle structures here are your antigens which are attached on the surface of your RBC, we will add a protein serum, we will add an antibody which will react with the antigen present in the RBC and it will cause an agglutination or cell clumping. Okay? So if an antigen present in the blood of the patient will be mixed with a matching antibody, it will lead to agglutination or cell clumping as seen in this image. So that is how we will know that there is a reaction or a positive reaction. So here is an image that will allow us to be able to understand this. If your blood type is A, then your RBC will have A antigen found on the surface of your RBC. Again, if your blood type is A, your red blood cell will have A antigen found on the surface. That is why we said that the antigen gives the cell its identity. It is the one that determines your blood type. Okay? If your blood type is B, then your antigens are of the B antigen. If your blood type is AB, it means that you have both the A and the B antigen present in your red blood cells. And if your blood type is O, then it means you have none of those antigens. No A antigen and no B antigen. That's why it's 0 O blood type. As mentioned, in our plasma, there are also antibodies present. And usually, the antibody present in your blood will be the opposite of your blood type. Okay? The antibody present in your blood will be the opposite of your blood type. Let's take this first one as an example. If your blood type is A, you will have A antigen expressed on the surface of your RBC. But in your plasma, it will have the antibody of the opposite blood type, meaning it has the anti-B antibody. Okay? It has the anti-B antibody. It cannot be an anti-A because if the antibody present in your plasma is still A, then your anti-A antibody will react with your A antigen and will cause clotting or cell clumping inside your vessels, and that could be very dangerous. That is why the normal antibodies present in your blood are of the opposite blood type. Let's go to blood type B. Since for blood type B, their antigens are of the B antigen, then the antibody should be of the opposite blood type, meaning what they have are anti-A antibody. For AB, they have both the A and B antigens. So there are no more opposite blood types. So there are no antibodies present in their plasma. No antibodies present in their plasma. While for group O, there are no antigens present, no? They have no A or B antigens. That is why in their antibodies, they have both anti-A and anti-B antibody. Alright? Hopefully that's clear. Now, let's do some blood typing. So in blood typing, we prepare a glass slide. This is a glass slide, and we put 3 drops of blood in each one of them. So 1 drop of the patient's blood, another one here, and another one here. What you see on top are the vials or serum that we add on to each individual drop of blood for us to determine the blood type. So the first chemical that you'll put is the anti-A serum. What is inside the anti-A serum? It has the anti-A antibody. Okay, it has the anti-A antibody. So it will try to detect if you have the A antigen. Okay? Kasi remember, the A antigen will react with the anti-A antibody to produce clumping. So in order for you to remember that, the first specimen or the first chemical is color blue, and that blue is anti-A. I put here azul. Anti-A serum is azul. For the next one, we have the anti-B antibody serum, which is yellow colored. So for us to easily remember, anti-B, banana colored, no? So what this does naman is to detect if you have the B antigen in your blood. So if your blood type B, then there will be clumping or agglutination in the glass slide. And for the last one, we have your anti-D, which is colorless. Anti-D naman is what tells us, well, it's the one which tells us if the blood is positive or negative, right? I'm sure you've heard of O positive, AB negative. So the positive negative is based on your anti-D. which is colorless. Okay? Now, putting everything together, let's say that we have a glass slide here with 3 drops of blood. We place 1 drop of your anti-A, which is azul colored, to the first drop of blood. We put 1 drop of your anti-B or the banana colored vial in another drop of blood. And lastly, we put the colorless anti-B in another drop of blood. We'll try to mix this one and this one and this one. And you'll be able to get this, no? Let me explain this and please do listen. So for the first one, if your blood type is AB, then what antigens are present? You have antigen A and antigen B. So remember that these 2 drops of blood both have antigen A and antigen B. For the first one, we put anti-A antibody. So there was a reaction or clumping. As you can see, no, nag-clump yung blood. There was clumping of blood because anti-A found in this vial reacted with your A antigen found on the surface of your RBC. And for anti-B, there was also a reaction because anti-B here reacted with your the anti-B here reacted with your B antigen. Kasi your type is AB, so it has both A and B antigen. So this is how a positive result looks like. Since there is a result here, we can say that there is A here, A antigen. And here, there's also reaction, clumping. We can say that there is a B antigen here. Therefore, the blood type is AB. For the second one, type A, if your blood type is type A, then you have the A antigen. So once you put anti-A onto that drop of blood, there will be clumping. Therefore, there is A antigen here. For the second one, even if you put anti-B, there is no reaction because normal plasma of type A blood has anti-B antibody. Remember? So type A has A antigen and anti-B antibody. So it already has its natural anti-B. You add more anti-B, there will be no reaction because they will just join together. For type B, same principle. You have your B antigen here in this type of blood. You add anti-B, there will be clumping. And for anti-A, there will also no reaction because type B blood already has its natural anti-A antibody. And lastly, for type O, there is no reaction, mainly because the cells in type O do not have any type of antigen. Okay? It only has both type of antibody. So here is an exercise which I want you to answer. Okay? So let's start with number 1. Please do not include this first cell here. No, this is your control lang. So let's just focus on the second, the third, and the fourth. Again, if there's a reaction at the fourth, you say positive. So for the first one, there's a reaction with anti-A, meaning this blood has your A antigen. So A, there's no reaction here, and then there's another reaction here, so positive. Final blood type, A positive. Ganun lang siya kadali. Number 2, there's a reaction at B, no reaction at A, so B positive. Okay, so that's how you will do this exercise. For the third one, we have reactions in all 3 panels. So this is AB positive. For the fourth one, no reactions in all. Therefore, there is no A, there is no B, and there is no positive. So this is O negative. O negative. Number 5, no reaction in both A and B, but there's a reaction at the end. So this is O positive. O positive. 6, 7, and 8, I'm sure that you can already do this. Number 6 is A, B negative. Number 7 is B negative. And number 8 is A negative. Now, the next one is preparing a blood smear. A blood smear is also one of the bread and butter of medical technologists. So, it's quite hard to teach this unless you do it talaga. Anyway, a blood smear is prepared to get a general picture lang of the blood. So, when you do your smear, it should be covering 3/4 of the entire slide. It should be tapering at the end or feathering. There should be a feathering at the end. And when you stain it, it should not be too dark colored. But in this image, it's just perfect. No, this is the one that is not yet stained, and this one already was stained. So here is a blood smear example. Please do take note of this because you will draw this on your lab manuals. Next, we have your differential WBC count. So what do we do here? We look at the blood smear we prepared and we try to examine it in this way, no? From one side to reach the other side, and then we will go down until we're able to count up to 100 white blood cells. We count 100 white blood cells. And the goal is to get the relative proportion of the different WBCs. Because if one of them is too elevated, then this can tell us that there is an infection inside the body, whether it be viral, bacterial, or even parasitic or allergic, no? So this will tell us the type of infection. So after counting to 100, the machine will stop on its own. It will show you how many neutrophils, how many lymphocytes, eosinophils, monocytes, and basos you were able to detect. I think this is the second to the last one. This is the hematocrit test. So what happens is we put a sample of blood in a very minute, very thin tube. And then that tube is left to stand. And then we're able to determine how many percent of that blood is composed of your plasma or serum, and how many of those blood is composed of your formed elements. Okay, because remember, your formed elements must only constitute around 40% of the blood. If the formed elements are more than 40, then we can say that the patient can be dehydrated. Kasi we try to determine the volume of your packed cells. But if it is too fluidy naman, we can say that the patient is overhydrated. So for 2MTO1 and 2MTO2, these are the pages that you need to draw. 343, the blood smear drawing only, and page 344. Deadline of your lab reports is on Sunday, 10 p.m. Okay? So that's it for the blood. If there are any questions, feel free to message me in my email. Okay? Thank you and God bless.